Now this is the second part of the epigenetics lecture, and in this we're going to cover the histone. We've previously covered DNA methylation, so silencing of gene promoters and epigenetic changes post-fertilization. What we're now going to cover are histone modifications, looking at how histone changes can affect whether chromatin is genes present on a region of chromatin can be expressed or not. So firstly, let's take a look at the structure of chromatin. And if we look at individual chromosomes within a cell, chromosomes are made up of DNA and histone proteins. And if we work backwards, here's the DNA sequence. You're all familiar with the double helix of DNA. Now DNA is wrapped around histone proteins, and we call these nucleosomes. So we've got 8 proteins in this sphere here and another histone protein there, and the DNA wraps around each of these spheres a couple of times and then wraps around another one and then around another one. And if we look at the number of nucleotides from this point here to the same point on the next histone core or nucleosome, is about 200 base pairs of DNA. So when we actually work out the mass of chromatin, it's approximately half of it is DNA, half of it is histone proteins, and the DNA is predominantly wrapped around histone proteins, and then there's these regions of linker DNA that are not wrapped around histones. Now this means that these histone proteins can actually control the likelihood of transcription of DNA by making modifications to these histone proteins, and this can fine-tune gene expression, usually coupled with DNA methylation as well. So this slide shows what happens if you take some chromatin and break it apart. Now we refer to these as nucleosomes, which is DNA wrapped around a core of histone proteins. So the DNA wraps around like this, and as I mentioned earlier, from that point to that point is 200 base pairs of DNA, and there is a core of histone proteins, and this is what chromatin is like. If we were to digest this with a nuclease which degrades this linker DNA, you would end up with DNA wrapped around a histone core, and then if you disassembled all of this with a high salt concentration to break away the RNA, sorry, the DNA from the protein, you would find there's 147 base pairs of DNA, and there's an octameric histone core, and if you were to further break that apart, you've got 2 histone 2As, 2 histone 2Bs, 2 histone 3s, and 2 histone 4s. Now if we look at what that looks like in terms of DNA, well, the DNA is not always wrapped around the histones, and so this shows DNA tightly wrapped around the histone, but this region here shows a sequence where a transcription factor might bind to. So for example, you will have come across you'll come across steroid hormones and steroid hormone receptors. So if you take the example of testosterone, testosterone binds to the androgen receptor, and the androgen receptor recognizes a very short sequence of DNA that we call an androgen response element. So the androgen receptor bound to testosterone, when it's bound when testosterone is bound to the receptor, it can then recognize a short sequence of DNA, which might only be 8, 10, 12 base pairs in length. So this could represent something like the androgen receptor binding to an androgen response element. Now the only way it can bind is if this DNA unhooks from the histone transiently to allow proteins to bind to the DNA, otherwise proteins can't bind to this DNA. So if the DNA is tightly wrapped around the histone, transcription factors can't get in. If the DNA is loosely wrapped around the histone, transcription factors can get in, and that can help to activate gene expression. Now where we're going to here is modifications to these histone proteins determine how tightly DNA is wrapped around the histones. So we can make modifications to histone 3 in particular that means DNA is tightly wrapped around here, and that silences gene expression, or we can do other modifications such as acetylations, and they will make the DNA much more loose, and therefore transcription factors can bind. And this is summarized quite nicely here. This shows the histone octamer core, showing DNA wrapped around it, and sticking out from these core proteins are what we call the histone tails. And these histone tails are regions of protein, the N-terminus of the protein, which sticks out a little bit, and this is what becomes either acetylated or methylated. And the simple rules are, with histones, if you acetylate this histone tail at a very specific place, that will make gene expression more likely because the DNA wraps much more loosely around the histone core, whereas for most methylations, that will make the DNA wrap very tightly around the histone core and silence this chromatin, preventing gene expression. However, just to complicate things a bit, there are some methylations which are activating. So all acetyl modifications to the histone protein are activating. Most methyls are inhibitory, but some methyls are activating, just to complicate things. So this is summarized here. We have chromatin. Let's start off with chromatin in the unmodified state, and this is histone H3 tails sticking out unmodified. If we acetylate those, we can, and that would be acetylated with an enzyme called histone acetyl transferase. That will add acetyl groups. We'll get lots of acetyl groups, and that will result in gene expression being potentially switched on. Because that's allowing transcription factors to get in, and then RNA polymerase can get in, and then we can get transcription. However, if we remove these acetyl groups from this acetylated histone to non-acetylated, that is done by an enzyme called histone deacetylase. Histone deacetylase will take the histones back to the non-modified form, and when they are non-modified, then histone methyl transferase can add on a methyl group, and that can then recruit what we call a heterochromatin protein and cause the chromatin to be condensed and silent. So these two enzymes are really crucial. Histone acetyl transferase makes chromatin active. Histone methyl transferase generally makes chromatin silent. And this is summarized here. Now this is all relating to histone H3. So there are other histones, but histone H3 is the one that is modified the most. And this little sort of quadrant here represents the core of the histone, and then this represents the N-terminal tail that sticks out. So over 40 amino acids stick out as a tail out of the histone core, the nucleosome, I should say. And all of these sites here can be modified by a post-translational modification. So a post-translational modification is where you've got the translated protein that is produced, and then you can start modifying those amino acid residues with different groups. So these numbers here represent the amino acid from the N-terminus. So an R is arginine and K is lysine. And it's the lysines that are particularly relevant here. This just shows all of the potential sites that can be modified with either an acetyl group, a methyl group, or that's a phosphate. And the really important ones that we're interested in is lysine 9 methylation, and that will cause gene silencing. Lysine 9 acetylation will cause gene expression, and lysine 27 methylation will cause gene silencing. Now this is the one that's a bit of a problem. This is lysine 4 methylation, which also causes gene expression. So this is the only methylation that causes activation of gene expression. So we have some codes that we use in the sort of epigenetics trade, and we'll see H3K27, and that refers to the amino acid lysine 27 of histone H3, and then we'll follow that up with maybe Me3 for trimethylation. So you can have, so this is lysine, this is the side group of lysine, and there's an NH3 group on there. Normally it can be acetylated, so this is an acetyl group, and then it can be trimethylated, so this is 1, 2, 3 methyl groups added to it. So if you see the code H3K27Me3, it means that this has happened, which is this silencing of histone H3, and that's particularly relevant to X chromosome inactivation, and also this fascinating group of proteins called Hox genes, and these control embryonic development. Now, much like DNA replication maintained the 5-methyl cytosine methylation pattern, when chromatin patterns are copied during DNA replication. So here we've got some DNA wrapped around histone cores in a cell. When that cell undergoes DNA replication, some of the histones will go one way, some of the histones will go the other way. So you'll end up with DNA here, which has got histone modifications on some of the histone cores, but not the others. Now, the enzymes histone methyltransferase and histone deacetylase and histone methyltransferase and histone acetyltransferase will come along and basically read the histone patterns on the nearby histone cores and find any unmodified histones and fill in the gaps, so that the histone pattern here is matched in both of the daughter cells when that DNA gets replicated in S phase, and then subsequently we get cytokinesis after M phase. The histone pattern here will be matched with the histone pattern in both of those daughter cells, so that the histone pattern is inherited into the daughter cells. However, even though that it is inherited, these patterns are reversible under cellular differentiation. So if a cell turns into a different type of cell, then these histone tags can be stripped off and changed to change the behavior of that cell. Now, it's really clear how this happens in X chromosome inactivation. So in biological females with 2 X chromosomes, so 46XX, in each cell there are 2 X chromosomes, one of which is silenced, and that is silenced by this modification here. So here we can see lysine 27 is methylated in X chromosome inactivation. So the whole chromosome is effectively silenced by histone modifications, and that completely changes that chromosome, and that chromosome is visible as a bar body, it's called. It's little, in cells from females you can often see this little condensed chromosome, which is a condensed inactivated X chromosome because the histone pattern has been completely changed. Now, this whole process is kicked off by a gene called the X-ist gene, which maintains only one active X chromosome in any one cell. So if you're 46XY, this gene is switched off. If you're 46XX, this gene is switched on. And if you're 47XXY, so an extra X chromosome, it's switched on to make sure only one X chromosome is switched on at any one time. Now, if you're interested more about X chromosome inactivation, it's worth taking a look at this particular link here. And this is from a really good site, which is called Citable, from Nature Publishing Group, Nature Education. Some really good articles on there about general biology and genetics in particular. But in this article, it explains all about inactivation of X chromosomes, lionization and how it was first identified back in the 1960s, and then it talks about the exist gene or the exist RNA that is produced, which effectively coats the chromosome with this RNA and then silences that particular chromosome. So if you follow that link and have a read through, there's some really quite complicated but interesting stuff about how the X chromosome is inactivated. Now this process is strangely relevant to anyone who's got a tortoiseshell cat, because a tortoiseshell cat is coloured in this way because of random X inactivation during embryonic development. So what's going on here is we've got the maternal X chromosome and the paternal X chromosome carrying genes for different fur colour. So on the maternal X chromosome there's the gene that gives the gingery colour fur, and on the paternal there's the a gene that is giving the black fur colour. During embryonic development you get random X inactivation within individual cells, and then you get like a patchwork of some cells expressing black, some expressing ginger, and when we look at the adult, they are what we call a mosaic. Some cells express the black gene, some only express the gingery gene. That's because either the paternal X chromosome is switched on, let's say here, and the maternal X chromosome is switched on in these gingery regions. So in summary, epigenetics refers to changes to DNA and chromatin expression caused by changes that don't alter the direct DNA sequence. So we have 5-methylcytosine, which affects the cytosines. It's still a cytosine, it's still coded as a cytosine, but it's modified in such a way that it can silence gene expression. We can have histone modifications, particularly methylations, which mostly silence chromatin. Acetyls on histone H3 will make chromatin more likely to be expressible, or the DNA be expressible. The main changes that we will see is histone H3 lysine 27 Me3 is a strong silencing tag, particularly involving X chromosome inactivation. Histone H3K9 Me3 is also silencing, whereas histone H3K9 acetyl is activating and creates looser chromatin, and that looser chromatin allows access of transcription factors, and then that chromatin is much more likely to be transcribed. So we say that the chromatin is active, and that is the formation of chromatin, which is euchromatin, whereas heterochromatin is tightly methylated and compact, so gene expression cannot occur from heterochromatin. Now there are a few bonus slides if you're interested. This slide talks about how the X chromosome is inactivated, and it's taking images from that citable article that I've just shown you. And then there's a couple of slides which are really quite hardcore relating to imprinting, and these are imprinting. It relates to regions of the genome which are not demethylated post-fertilization, so that you only get paternal expressed genes and maternal expressed genes. So these 4 genes are only expressed from male-derived DNA, these are only expressed from female-derived DNA, and then if you have some genetic abnormalities, such as a duplication of the chromosome on here, you can end up with an absence of either these genes being expressed or these genes expressed, which is sort of explained by this slide. However, this is a really difficult concept. If it's tipping you over the edge, maybe you want to skip the imprinting. It's not something. It's there as a set of bonus, interesting slides if you are interested. But if you are interested, these genetic abnormalities, so deletion of this region, will result in either Prader-Willi syndrome or Angelman syndrome, and these are known as imprinting diseases. Okay, that's probably enough on this topic. So the things that I want you to really understand from this particular lecture is, if you can understand how DNA methyltransferase and CpG island methylation silences gene expression, that's good. And if you can explain how histone modifications either activate chromatin so that genes can be expressed or genes can be silenced, that is the key set of concepts that I want you to understand.